Alright, this week we are going to study bone and blood in the lab. So this begins with a continuation of chapter 5. So first we're going to talk about bone. This is a cross-section through the diaphysis of a long bone. This particular section was prepared using a method that's called decalcified bone preparation. You do need to know this, be able to identify what technique was used to prepare this slide if we show you this. So decalcification is when the bone is treated with acid, which helps to remove the hard calcium and phosphate salts, which essentially softens the tissue, making it soft enough to where you can section through it and then stain it. So if you see a section of compact bone that has been stained with H&E and giving it a pinkish color, as you can see here, then you know it's been prepared through decalcification. Okay, and so what you can see with this low magnification view of the bone, you have the outer part of the bone, which is composed of compact bone, and then you have the inner marrow cavity, which consists of blood, hemopoietic stem cells, adipose tissue, and some other cell types. There is a small amount of cancellous or spongy, also called trabecular bone, that sometimes is found close to the marrow cavity at the superficial part next to the compact bone. Long bones are mostly consist of compact bone, however. So here's a higher magnification view of the outside portion of this long bone. Again, here's the marrow cavity seen at a higher magnification. All of these red cells that you see here that are densely packed in there, those are red blood cells or erythrocytes, so there are lots of those. These large white cells are adipocytes. We're going to come back and take a closer look at this later on when we talk about blood and show you some more specific cell types that can be found in here. For now, you should focus on this part of the bone, which is the compact bone. And you can see that it is surrounded by a layer of connective tissue, which is called the periosteum. The periosteum is composed of a dense, irregular connective tissue and also contains some osteoprogenitor cells that give rise to osteoblasts and osteocytes. This region, the periosteum, is analogous to the perichondrium, which surrounds cartilage tissue. So here's the actual compact bone structure, and the advantages of preparing this section with decalcified bone preparation is that it spares or retains the living part of the tissue, which are all of the cells. So you can see compact bone is riddled with little pits or lacunae, just like within cartilage, because again the matrix here is solid, and within those lacunae there are cells. Now at this low magnification it's difficult to see those. However, you can also see there are larger openings or canals. Some of these are relatively circular, some of them are longitudinal to the long axis, perpendicular to the long axis of the bone, or at an oblique angle. And these contain blood vessels and nervous tissue and connective tissue that help to carry blood or allow signaling to occur between cells of the living bony tissue. The disadvantage of this particular preparation is that it really destroys the inorganic architecture of the bony matrix, and so by removing the calcium salts and sparing the cells with this method, when you section it, you don't actually see a lot of the geometry of the bony matrix. And in order to see that, we use an alternative preparation, which is called a ground bone preparation, which we'll show you here in a little bit. So this slide shows a higher magnification view of compact bone prepared by the decalcification method. And you can notice that there are these circular canal structures. Some of them are large in diameter, like these. Others are smaller in diameter. These are called central or haversian canals. And again, these are often filled with blood cells that you can see here because there are blood vessels and nervous tissue that pass through here. The reason that you have different size haversian canals is that the younger the canals are, the larger in diameter they are. But as this bony tissue ages, the matrix becomes remodeled, and so you have new bony matrix laid over some portions of this canal which was exposed before, such that you get older haversian canals or central canals, which are more narrow in diameter. So the smaller the diameter, the older the central canal is. The central canal or haversian canal is a center part of a larger structure which is called an osteon, and an osteon consists of several rings of what are called concentric lamellae that are arranged together. These ring-like arrangements of the bony matrix are really hard to see with a decalcified bone preparation, and it's easier to see the overall structure of the osteon later on when we look at ground bone preparations. So in addition to these round-shaped haversian or central canals, you have these other canals which are roughly perpendicular or oblique to those. These are called perforating canals or Volkmann's canals, and these carry blood vessels in from outside of the bone to supply the bone with blood. So these blood vessels will travel through these perforating canals from the outside, and then will join with these central canals or haversian canals, at which point these blood vessels branch off and travel along the longitudinal axis of the bone. Here's an even higher magnification view, and here's a small central canal or haversian canal, which is from an older osteon. And if you look carefully, you can actually see these ring-like structures in the bony matrix here. Again, they're not as clear as when using a ground bone preparation, but you can begin to see the circular nature of these concentric lamellae. What you can also see at high magnification are the osteocytes, which are the cells that help to maintain the bony matrix. And because that matrix is solid, these osteocytes are found deep within these lacunae. Later on, we're going to show you cells that give rise to osteocytes, which are called osteoblasts. And so osteoblasts initially lay down bone that's newly being formed, and they're analogous to the chondroblasts that initially produce the cartilaginous matrix of cartilage. And likewise, the osteocytes are maintaining the bony matrix, and so are analogous to the chondrocytes that maintain the matrix of cartilage. So those structures can be easily seen in decalcified bone preparation. This section here is also of compact bone, but you can see it has quite a different appearance because it was prepared in a very different way. This method of preparing bone that you need to know is called the ground bone method, and essentially it involves grinding a section of bone until it becomes very thin, enough to where light can actually pass through it. And so that's it. Then it's basically mounted on the slide, and you can look through it with the scope. Because the calcium salts and other salts are maintained in the matrix, the architecture of the matrix is preserved really well. So it's too hard to stain it. You can't stain it because the tissue is not soft enough. But it does preserve the architecture much better. One of the disadvantages, however, is that it kills all of the cells in this tissue. So when you look at bone tissue that's been prepared in this method, all the cells have been dead, have died before placing it on the slide. And so there really aren't any osteocytes visible here. They've all been replaced, or they've all basically died and been replaced by trapped pockets of air and bone dust and things like that. So this is a low magnification view of a compact bone that has been prepared by this method. And there are two types of lamellae that you can see on the slide. In addition to the concentric lamellae, you have a inner, what's called circumferential lamellae, that surrounds the marrow cavity. So this is where the marrow cavity would be in the long bone. But again, you don't see anything here because all of that soft living tissue has been dead. And these lamellae here, instead of being part of an osteon with a relatively small diameter, these lamellae traverse the entire width of the bone here on the inner circumference. So these are called the inner circumferential lamellae. These lamellae here on the outside are even longer or larger in diameter, and they span the entire external circumference of the bone, and so they're called outer circumferential lamellae. Here's a higher magnification view of the inner part of the bone, and here we can see the actual shape of the osteons here. So this is a single osteon, this structure, and we can see it's composed of multiple concentric ring-like structures or lamellae of bony matrix. These are called concentric lamellae, and so you can count these different rings here. At the interface between concentric lamellae is where you find the lacunae that hold would normally contain the osteocytes. In this particular case, these lacunae are all empty and trapped with air and bone dust, so they appear relatively black in appearance in this particular slide. In some slides, depending on the amount of dust or air that's trapped, they may appear either dark or light in coloration. So here we can see lacunae, these dark spots, and you'll see that the lacunae are at the interface between adjacent concentric lamellae. So this is a concentric lamellae on one side of this lacuna, and on the other side of this lacuna you have another concentric lamella. So the lacunae sit right between where adjacent concentric lamellae face each other. Right here in the middle of this osteon is the central canal or Haversian canal. Haversian canal is a name, it reflects an older naming system, and so another name for an osteon is a Haversian system. For the purposes of this lab, you should be able to know both terms, and if you're asked to write that on a quiz, you can use either form. The third type of lamella, besides circumferential lamellae and concentric lamellae, are called interstitial lamellae. And interstitial lamellae are irregular shaped lamellae in between the concentric lamellae. So here we can see lots of interstitial lamellae. Notice that they're not circular shaped. Often they're kind of flat shaped or arch shaped, and they are found at various angles. These form as bone remodels over time, and the positions of the osteons change a little bit. And so in the interstitial spaces between the concentric lamellae, you have these odd-shaped interstitial laminae. Here's a higher magnification view, again, of a single osteon here. You can see the structure of the lacunae at really high magnification here. And at this magnification, you can easily see lots of these little thin, would appear to be spidery shaped or filamentous black structures here. These are actually very tiny canals that are appearing black because, again, they've trapped bone dust and air in them. And the name for these tiny little canals are called canaliculi. And so canaliculi connect multiple lacunae here, and they provide a communication network by which individual osteocytes trapped within their lacunae can communicate with one another. So in cartilage, these canaliculi aren't really necessary because substances can diffuse slowly through the solid matrix of cartilage. But in bone, the matrix is very hard, and there's not really any diffusion that occurs through that bony matrix, and so the cells can instead communicate to each other through this series of interconnected canaliculi. Note that this network of canaliculi also contains cytoplasmic processes of cells, and so individual cells, parts of their cytoplasm may be able to directly contact each other through these canaliculi. So as you can see with this type of preparation of bone, you observe the bony tissue in a very different way. And so in reality, bone actually does have this very intricate extracellular matrix, but it also is filled with lots of living cells which are preserved using the decalcification method. Here's another view of compact bone that has been prepared using the ground bone method. Here you can see a Volkmann's canal or perforating canal. Here's another one. Okay, so again, these are kind of elongated or cigar shape as compared to the central canals within the osteons here. Some of them look darker than others, some of them are lighter. On your particular slide, whether they're dark or very light in the center really depends on how that particular slide was prepared. So moving along, this is a section of bone from a skull developing flat bone, and this slide reflects one of the ways in which bone can develop. This type of development is called intramembranous development. That's how flat bones form. You can see that this bone is stained, which means it's been produced by decalcification methods. So this is a decalcified bone that's been stained with H&E. It's important to remember that the matrix of bone stains eosinophilic, and so this eosinophilic structures that you're seeing here is the actual bone, not the white stuff. The white stuff are the spaces in between the bone. Bone, as it initially forms an immature type of bone called woven bone, which is very similar to spongy bone or trabecular bone. And so it has lots of holes in it, kind of like Swiss cheese, and the holes are the whiter, paler areas, and the dense bony matrix, which in this type of bone are composed of trabeculae, are the eosinophilic structures. So it's important to remember that it's not always necessarily counterintuitive, because sometimes you want to think of bone as being light or yellowish in color, and that's only true when you're looking at bone that hasn't been stained. So here's a higher magnification view of this developing trabecular bone. Again, this type of bone gets its name because of the name of the structures, trabeculae, which are these elongated finger-like structures. And you can see that these trabeculae are riddled with lacunae, and inside these lacunae are osteocytes, just like in compact bone. Here's a high magnification view, and here's a kind of a hook-shaped trabecula right here, and you can see there are lots of lacunae with osteocytes inside them. Note that the organization of the matrix is different than compact bone here, and that trabecular bone does not have central canals with concentric lamellae or anything like that. It has a very different organization. In addition to the osteocytes within the lacunae, there's a different type of cell that you can see that sits on top of the trabecular bone. Because this bone is in the process of being developed, you can see the osteoblasts, which are laying down the bone. So these cells, again, are called osteoblasts, and they always sit on top of the surface of the trabecular bone. Osteocytes are trapped within lacunae, both in trabecular and in compact bone. So recall that osteoblasts are the cells that are laying down new bone. After they lay down bone, they get trapped within the bony matrix and are found within these lacunae. So osteoblasts on the surface of the bone give rise to osteocytes, which are located in lacunae. Now, in some sections, you may see osteoblasts appearing to sort of hover over the surface of these trabeculae. This is an artifact of slide preparation. In fact, they normally always sit on the surface. They wouldn't be found like this. But because they're on the surface and there's nothing to anchor them down, when this tissue is sectioned, a lot of times they get perturbed and actually moved off of the surface of the bone. There is a third type of bone cell which you need to know and be able to identify, and that's called an osteoclast. This cell right here is an osteoclast, and as you can see, it has quite a bit of a different shape from osteocytes and osteoblasts. Osteoclasts are much larger than the other two cell types. Like osteoblasts, they lie on top of the surface of the trabeculae, but they are much larger than osteoblasts. The shape of the cells is more irregular, and they are usually multinucleated, and they may have several or even dozens of nuclei. In this particular cell, you can see at least 1, 2, 3, 4 or 5 different nuclei in this one cell. Osteoclasts look dramatically different than the other cell types because they come from a different cell line. These particular cells are derived from monocytes, which is a type of blood leukocyte that we're going to look at in a little bit. So these are actually a type of specialized macrophage, and they actually cause erosions on the surface of the bone, releasing calcium back in the blood, and so they're critically important for calcium homeostasis within the blood. Because their role is to remove bone, so these are bone destroying cells, whereas osteoblasts would be bone synthesizing or bone producing cells, and osteoclasts would be bone maintaining cells. Because these destroy bone, they are commonly found near these surface depressions in the bone. These surface depressions are called Hauschup's lacunae. And this is one of the best places to try to find osteoclasts. Osteoclasts are far less common than are osteoblasts and osteocytes. You should have no problem finding those two, but there are not nearly as many osteoclasts. You really have to hunt around the slide to find them. If you can find these Hauschup's lacunae, or these depressions, you may often see osteoclasts nearby, although not always. So that's a good place to start looking. Here's another section of trabecular bone. In this case, you have multiple deep Hauschup's lacunae. Here's one here. Here's another Hauschup's lacuna. Here's another one over here. And you can see multiple osteoclasts that are multinucleated sitting right here on these Hauschup's lacunae. Moving along, this is a section of a fetal finger. So it's a tiny little finger here. You can see the three segments of or three phalanges of the finger here. So here's the distal phalanx, here's the middle phalanx, and here's the proximal phalanx. You can see the epiphyses of these developing bone are composed mostly of cartilage here, and the matrix of cartilage, hyaline cartilage, in this case, is more basophilic. It has a glassier appearance, so that's why the epiphyses of these bones appear that way, have kind of that bluish color to them. The matrix of bone, again, is more eosinophilic, and so the newly laid down bone in the diaphyses of these bones is eosinophilic. So these are long bones that are actively developing, and development of long bones occurs through a different process which is called endochondral ossification. So flat bones develop by intramembranous ossification, and the bony trabeculae develop directly from mesenchymal cells. This type of ossification, again called endochondral ossification, gets its name because the newborn bone, trabecular bone, is laid down from a template composed of hyaline cartilage. So this bone develops from hyaline cartilage, not directly from mesenchyme. So in the next slide, we're going to take a look at the regions of the epiphysis of the bone at the interface where the new bone is being laid down. And so there are different zones of endochondral development that you need to be able to identify. So this is a section of hyaline cartilage at the epiphysis of the bone, and this is often referred to as the zone of reserve cartilage. This is essentially just normal mature hyaline cartilage. So you can see chondrocytes within their lacunae, surrounded by territorial and interterritorial matrix. These cells are in reserve, and eventually these cells will begin proliferating and then eventually ossify and become convert the matrix around them to a bony matrix. So in the meantime, these are held in reserve, and so this is called the zone of reserve cartilage. If you look at this end over here, which is closer to the diaphysis of the bone, you can see that the cells are closer apart and are smaller, and that they are kind of found clumped together in stacks. This is called the zone of proliferation, which gets that name because the cells are rapidly dividing. That's why they are smaller and closer together. Again, this is zone of proliferation further down, even closer to the diaphysis, and then further down closer to the diaphysis, you can see the size of the chondrocytes have enlarged, as well as the lacunae that they're found in are bigger. This is called the zone of hypertrophy because the cells are increasing in size and hypertrophying. Even closer to the diaphysis of the bone, close to where new bone is being laid down, you have another zone which is called the zone of calcification and cell death. And that appears when you see a change in coloration of the matrix from a lighter basophilic color to a deeper, almost more purple basophilic color. And that's because the chemistry of the matrix is beginning to change in this particular area. Again, here you can see the transition between the zone of hypertrophy and the zone of calcification and cell death. It gets its name because these cells are secreting calcium salts into the matrix, which is beginning to turn the matrix hard. And as the matrix hardens, it prevents diffusion of nutrients, which ends up killing the cells. So as the matrix calcify, the cells also begin dying. And then finally, you have the final zone of development, which is called the zone of ossification and resorption. And you can see that where the matrix starts turning its mature eosinophilic color, which is typical for bony matrix. And so here you have osseoid and other substances that change the chemistry of the matrix, making it more eosinophilic in appearance. You can also see that the matrix here thins out. A lot of it starts being resorbed, which creates these bony structures called spicules. These are these bony projections, and then spicules eventually get converted into trabeculae, which is the basis for trabecular bone. In long bones, most of that trabecular bone then goes on to be converted into mature compact bone, which is a process we're not going to talk about in this lab. So here's a lower magnification view of this long bone that's undergoing endochondral ossification from a cartilage template. I'm going to go through these layers again real quick so you can see them all together. This outermost layer towards the epiphysis, here's the epiphysis out here, this is in the diaphysis of the bone. So this outer layer is the zone of reserve cartilage. Here you have the zone of proliferation. Here you have the zone of hypertrophy. Here you have the zone of calcification and cell death, where the matrix becomes a darker purple. And then here we start seeing eosinophilic matrix. This is the zone of ossification and resorption. You will need to know those for the practical, although I won't quiz you on those specific zones for the quiz yet, so just make sure you're able to identify those on your slides for the practical. So that's about covers it for bone. We're going to now talk about blood cells for a little bit, so you'll spend some time in lab looking at blood smears, trying to identify different cell types that can commonly be found in blood smears. So recall that blood smears are typically stained with Wright stain, which is similar to H&E, but it allows you to be able to identify white blood cells more easily. So most of the cells in a blood smear are red blood cells or erythrocytes, which are these disc-shaped cells here. You can't see a nucleus in these because these cells are anucleate, so if you're looking for a basophilic nucleus, you won't find them. In fact, the center of the cell is very thin because these cells are actually biconcave in shape, and because the center of the cell is so thin, there's very little cytoplasm to take up the eosinic stain, and so they appear very pale in the center frequently. So you should be able to see these all over the place, but the interesting things on the slide that you want to try to find are the different types of white blood cells. So this white blood cell in the center of the slide here is called a neutrophil. Neutrophils are the most common type of leukocyte. They get their name because the cytoplasm has neither greater affinity for basic or the acidic dyes, so chemically they are neutral and stain just very pale, neither acidic, neither eosinophilic or basophilic. They are often referred to as polymorphonuclear neutrophils because of the shape of the nucleus. And it might look like the cell has multiple nuclei. This is actually a single nucleus, but it has discrete lobes. On average, neutrophils have between 2 to 5 lobes to their nucleus. Most commonly, they have 3. This particular neutrophil has 4 discrete lobes. And it might not look like they're connected, but there are small bridges of nucleoplasm which actually connect these nuclear lobes together. So this is a neutrophil. Neutrophils are antibacterial cells that are commonly found in the blood. You should have no problem finding neutrophils at all. This cell is a more rare type of white blood cell, which is called an eosinophil. Now, neutrophils and eosinophils are both classified as granulocytes because they possess granules in the cytoplasm. However, the granulocytes of neutrophils are very hard to detect because they're chemically neutral. Eosinophils get their name because their granules are very eosinophilic, and so they stain this kind of deep red. These are the second most rare types of leukocytes overall, so you really have to hunt to find these, but I know not every slide will necessarily have them, but some of the slides in lab always do have them. So if you can't find one on your slide, try to look at a neighbor's, and if you do find an eosinophil, be sure to share it with the class so that other students have an opportunity to see it firsthand through the microscope. Another distinguishing characteristic of eosinophils, besides having coarse eosinophilic granules in its cytoplasm, is they have a typically large bilobed nucleus. So here you can see a large basophilic nucleus. It has two large lobes. Here's one here. Here's the other one here, connected by a thin bridge of nucleoplasm, giving it a characteristic kind of barbell or dumbbell shape to it. So the nucleus of eosinophils is usually easy to see and very distinct. This cell is a basophil, and basophils are even more rare than eosinophils. They are the rarest of the leukocytes, so you really have to look hard to find them. Again, if you find a good example of a basophil, please share it with your neighbors so they have a chance to see it. Basophils are the third type of granulocyte. So they differ from eosinophils in that their granules are basophilic, not eosinophilic. The nucleus is usually very large and irregular in shape, and is actually difficult to see because the nucleus is also basophilic, and so it usually gets obscured by the coarse basophilic granules. So if you can, besides looking at the color, you can also look at the shape of the nucleus. If you can see the nucleus easily, you know this is an eosinophil. Sometimes when you're looking at a basophil, it's hard to tell whether it's more purple or bluish or more reddish in color. But if you look at the nucleus, that's another important clue you can use to distinguish between those two cell types. So here's a composite image of 3 blood smears. Here you have a neutrophil, here's an eosinophil, and here's a basophil, so you can compare and see what they look like. Here's a blood smear that has a lymphocyte in it, and a lymphocyte is a type of agranulocyte because they do not have granules in their cytoplasm. Lymphocytes are very common. You should have no problem finding them. They are the second most common type of leukocyte overall, behind neutrophils. And so there aren't quite as many as neutrophils, but there are a lot more than eosinophils and basophils, so you should be able to find them. Lymphocytes come in a variety of sizes, so looking at their size is not the best way to identify them. This is a relatively small, perhaps medium-sized lymphocyte, and it's a little bit bigger than red blood cells. Some of them can be quite a bit larger, though, and so the size, again, is not the best method of distinguishing them. What you should look for is their characteristic large, regular circular shaped or spherical shaped nucleus, which is very basophilic. And the nucleus is so large in these cells, it takes up most of the volume of the cell, such that you usually only have a thin little rim of cytoplasm outside of the nucleus. That's the hallmark characteristic of lymphocytes. Here's another example of a lymphocyte. Again, you can see the nucleus here and a thin rim of cytoplasm. There are B lymphocytes and T lymphocytes. You cannot distinguish those in a blood smear. It requires more advanced histological techniques to distinguish between them. So using a right stain, they look identical. So don't worry about trying to distinguish between B cells and T cells. You can't do it using this method. Here's another blood smear showing the other type of agranulocyte. These are the third most common cells, so they're easier to find than eosinophils and basophils, but harder to find than lymphocytes and neutrophils. This cell is called a monocyte. They are most commonly confused with lymphocytes because, like lymphocytes, they don't have granules in the cytoplasm and they have a very large basophilic nucleus. However, monocytes are the largest cell types, so they're bigger even typically than the largest of the lymphocytes, and so you can see this monocyte is several times larger than these neighboring red blood cells. Also, the nucleus of monocytes is more irregular shape instead of being perfectly spherical. They are either irregular shape or oftentimes have this classic horseshoe shape. So that's very different from lymphocytes. Additionally, monocytes have much more abundant cytoplasm, so you can see a lot of cytoplasm here, not just a very thin rim as you see for lymphocytes. Here is a composite image showing a lymphocyte on the left, and here's a different monocyte. This one has a little bit of the U-shape or horseshoe-shaped nucleus, but it's kind of irregular shaped as well. So again, there's some variability in what those cells can look like. Make sure that you are able to identify and distinguish between all 5 types of lymphocytes as I've shown you in this PowerPoint. This is a section of bone marrow. So this is going back to the bone marrow slide. The only slides that we have that show you the bone marrow are the sections of the long bone that have been decalcified. So I would go back and look at those. Here you can see some trabecular bone in this slide. You may not have trabecular bone in your particular slides because there's not a lot of it in long bones. And this is the marrow cavity right here. These large white appearing cells, these are adipocytes. You should be able to recognize those by now. Here's a higher magnification view. You can see lots of, just tons of erythrocytes or red blood cells in this bone marrow. So this is composing the red marrow. Yellow marrow is composed of adipocytes. So this particular bone section has both yellow and red marrow. As most bones age, the red marrow is replaced mostly by fatty adipose tissue or yellow marrow. One of the cells you can see here that we haven't talked about yet, these are called megakaryocytes, and these are huge cells with very large nuclei. And that's really where they get their name, megakaryocytes, cells with huge nuclei. And the cells themselves are just quite big. They're almost as large as adipocytes, and they're many times larger than red blood cells. You can see how small these osteocytes within the lacunae look here for comparison. And we look at these cells because these cells produce platelets, which are important for clotting of blood. So platelets are formed by budding off of the surface of these megakaryocytes. To actually see the platelets, they're released into the bloodstream. So you should be able to see platelets all throughout your blood smears. And platelets are not cells themselves, rather they are disc-shaped fragments of cells that bud off of the surface of megakaryocytes. So they don't have nuclei, they have a little bit of cytoplasm, and they just look like these little cellular fragments. You might think that they are just debris or dirt on the slide, but these are actually platelets, and you should be able to find them all over the place. Here is, just for review, this is a lymphocyte, and right here we have another neutrophil. So that's it for this week. Make sure you are able to identify all of those structures, and that's it.